Here we are. And statistics. All right, so again, on this one, if you take a look, most people are pretty solid on this material. If you're down here, there's a couple of people who just seem really lost. If you're one of them, you know, it's getting late. It's getting late. You definitely need to, in fact, I'm going to talk a little later about kind of the big picture of grades, because some people are doing really nice. Some people are really not even close to passing. We got to talk about that. We can realistically take a look at what's going on for everybody at some point. Oops. All right, so now describe at least 2 ways synapses are cleared of neurotransmitters. Most people got this. What are ways that synapses are cleared of neurotransmitters?
Reuptake.
Presynaptic cell.
Yeah, so there's reuptake into the presynaptic terminal. What's another way?
Diffusion.
We'll say it again.
They diffuse.
Just diffuse out. And then we're going to talk about what happens to that, because obviously if you have neurotransmitter just diffusing out, it could cause mischief where it could burst into some other synapse someplace. So we're going to see some other cells in this nervous tissue that are going to be important for kind of maintaining the extracellular milieu and kind of being the little custodial staff to make sure that you don't have stuff floating around you don't want. What's another way we talked about?
Enzymatic breakdown.
Enzymatic breakdown. Sometimes you actually have enzymes that just start chewing apart the neurotransmitter once it's outside the cell, and that's what stops it. Shark! So, yeah, this is, I have no idea why these guys, there's like about 4 movies now about these stupid sharks. But if there was a shark falling out of the sky from a Sharknado and you were trying to desperately stay alive, what division of your nervous system would be firing off on all cylinders? Sympathetic. All right, so here we see, and most people got that. Actually, everybody got that, which is good. Nerves that deliver messages from your eyes and your ears. And this is where you need to know about, this terminology is important. We're going to be using it a lot. Afferent, these are the ones that arrive at the brain and spinal cord. So things that are delivering things, the sensory side are afferent. Efferent would be motor. Nervous nerves I just made up to be silly, but somebody bit. Reflexive nerves I also kind of made up and a couple of people bit. This one, adrenergic, you need to know this word. Adrenergic is used, we're going to use it a bunch as we keep going into our description of the autonomic nervous system. Plus it's used all the time in describing different drugs as you get further on in pharmacology and stuff. So what does it mean to be adrenergic?
Cholinergic with adrenaline and not epinephrine or epinephrine.
So not cholinergic is something else. Cholinergic is dealing with acetylcholine. So
it's epi and not epi.
Yeah, so adrenergic meaning like adrenaline, but adrenaline, which in our native tongue we call epinephrine. And so epinephrine and norepinephrine are the things that are what we call adrenergic, things that relate to epinephrine or norepinephrine, also called adrenaline or noradrenaline. Those would be under the adjective adrenergic, adrenergic receptors, adrenergic pathways, adrenergic this or that. Adrenergic agonists, adrenergic antagonists. Again, like asthma inhalers. like albuterol, are adrenergic agonists, things that turn on your sympathetic nervous system to dilate your bronchial passages so you can breathe in the moment. Again, adrenergic antagonists like the beta blockers that keep your heart from speeding up, to help control blood pressure. So a lot of drugs, common drugs that are prescribed, are adrenergic agonists or antagonists. Choose all that are part of the central nervous system. So it should just be the brain and the spinal cord. When you get into motor or sensory, then we're in the peripheral nervous system, PNS. I'll make a little picture that makes this a little clear. Sometimes people get a little confused about this stuff. All right. So, I guess a little more detail about CNS versus PNS. Again, CNS, we always talk, is the brain and the spinal cord. And then people get confused though, because like one particular neuron might actually have one foot in the CNS and one foot in the PNS. PNS is all the stuff that's outside, the motor and the sensory. So let's say you've got your hand here. Let's say here's your finger. And you've got some sensory receptor. We're going to be looking at sensory receptors today in our lab. And the sensory receptor has an axon that sends a message. There's actually this dorsal root ganglion, and then the neuron goes into there. So there's this one neuron here. And people get confused because they're wondering, well, is that neuron in the PNS or the CNS? And it's like, it's both, right? The cell body is outside, it's in the PNS. This big long axon here is in the PNS, but this side over here, this is in the CNS. Same thing with motor neurons. I could have a motor neuron, that has its cell body in the CNS, but sends the axon down to operate the muscles. So when CNS is kind of like a boundary, anything, maybe I'll use this little highlighter thing. Anything that is within the boundary of the brain and the spinal cord, if you are inside the yellow zone, that's CNS. If you're outside of the yellow zone, you're in PNS. And again, you can have an individual neuron that is got different parts in CNS and PNS. So kind of the same way, right? If I'm open it, you can't see it. I open up the window here. I have my hand outside the window. And my body is in the house, but my hand is in my backyard right now, right? It's one person, but my hand is out the window in the yard. My body is here in the house, just like I have this neuron here whose cell body is in the CNS, but axon is in the PNS. So that makes sense. Because again, people get kind of turned around there. While we're at it, we might as well give it a little more terminology, just because it's going to be important as we start describing stuff. So, and the terms are different if you're looking at the CNS versus the PNS. So like if you have a collection of axons running together, a collection of axons If you're in the CNS, we're going to call them axon tracts. And we'll look at a variety of different kinds of tracts. We'll talk about commissures and projection tracts and things like that. But basically, when you have like a bunch of axons running together in the CNS, we call them tracts. In the PNS, what do we call a bunch of axons running together like a big cable?
A plexus?
Nerve.
It's a nerve. You know, when we talk about the ulnar nerve or the cranial nerves or the phrenic nerve or the sciatic nerve, all of those are just collections of axons that are running together like a big cable. So a nerve, the official definition of a nerve is a bunch of axons running together like a big cable. In the PNS. So collections of neuron cell bodies. In the CNS, they get a weird name. They're called nuclei. Again, it's unfortunate that that word is used for lots of other things, but we're going to be talking about, you know, the caudate nucleus and the lentiform nucleus and this and that nucleus. These are little sections in the brain and spinal cord that are collections of neuron cell bodies. In the PNS, we call them ganglia. Single is ganglion. So we'll be using both of these, or all of these terms at different points as we talk about the nervous system. You know, back to this picture here, you know, this would be like part of a nerve, you know, running towards my hand. If I have a bunch of axons that are outside the CNS that are running along, those would be part of a nerve. Cell bodies here that are outside of the CNS, that would be a ganglion. Meanwhile, you know, when I have axons running around inside the CNS, these are tracts. When I have collections of cell bodies in here, these are ganglia. So just kind of putting, yeah, you should know that terminology. If you've been in anatomy, you're like, of course I know that, I learned that. But if you haven't seen that or you don't remember it, we will be using those terms. Okay, what else? We just need to do a little more background information on nervous tissues so we have the vocabulary to talk about everything. Again, physiology is very intertwined with anatomy. So unless we know the basic anatomy, we can't really talk about the physiology. So let's just talk about nervous tissue. We're going to do this much more briefly and kind of superficially than we would in anatomy, but just enough till we have the basic parts and pieces. So let's talk about the cells. What are kind of the rock stars of the nervous tissue? Which cells are like the most famous cells that make up your nervous system?
Neurons?
Neurons. You know, sometimes we call them like nerve fibers because they're really long, or nerve cells. So neurons, these are the ones we've been talking about, but they aren't the most numerous type of cell in your nervous tissue even. Like over half the cells in your brain are different cells. They're not neurons. So we are going to talk a bunch about neurons, but it's important to pay attention to the other cells we call the glial cells. Glia means glue. Like in the old days, they used to talk about supporting cells in the PNS and glial cells in the CNS. Now they just kind of say glial cells for everything, makes life easier. Glial cells, also called like neuroglia. These are the cells that help support the neurons, but are not neurons. I should mention neurons come in different forms, which we don't need to know so much for this class. These are, if you've been in anatomy, you remember multipolar and bipolar and unipolar. I might actually mention some of those sometimes, but don't worry about that. You're not going to be tested on different subtypes of kind of neuron morphologies. So let's talk about the glial cells, the cells that aren't neurons that are important. The glial cells are different if you're looking at the CNS versus the PNS. So let's do that. Actually, let me do this a little differently. So in both cases, in both the CNS and the PNS, you're going to have glial cells whose job it is to make the myelin sheaths. So in both cases, we're going to have something that makes myelin sheaths. In the CNS, they're going to be oligodendrocytes. In the PNS, they're going to be Schwann cells. Schwann cells also have like they have a more modern name like neurolemocyte, but don't worry, you just call them Schwann cells. So oligodendrocytes versus Schwann cells. They're both the same in that they make myelin sheaths. Myelin is actually just a specialized type of cell membrane. Both of these work by, let me try here, hold on one sec. All right, so I should probably turn off my virtual background so this is easier to do. So I just made a cell. This is, the plastic is my cell membrane. The water in here is the cytoplasm. This little tangerine is supposed to be the nucleus. So this would be, let's say this is a Schwann cell. And what it does is it finds a neuron. Let's say the neuron is my arm. And it goes and it starts wrapping around and around and around until the whole, it actually goes around many times. Here I don't have enough, but you can see like the cytoplasm and the nucleus, they're still there kind of squished on the outside, but the cell membrane has now wrapped around like a jelly roll around my arm, making this insulation.
Is it one cell per like sheath, like, you know,
it
circle?
depends on whether you're in the CNS or the PNS.
Okay.
Let's take a look at that actually right now. So if you are in, let me actually dry off my iPad. So Schwann cells, it is one cell per sheath. So if I have my axon here, each Schwann cell is one of these sheaths. Here's like the nucleus here. Here's like the jelly roll of membrane around there. So each Schwann cell makes up one sheath. Schwann cells can also play a role in just keeping unmyelinated fibers from shorting out, right? Even if you have an unmyelinated axon, remember we talked about some of the slower neurons have unmyelinated fibers, but you still don't want them to touch and cross, have a short circuit across each other. So you can actually have Schwann cells play a role in actually just being insulators, holding unmyelinated fibers from touching each other. So that's another role that Schwann cells play. And then one last role that Schwann cells play that is pretty cool is helping repair damage. This idea of Wallarian degeneration. Let's say I have some neuron. And again, it's got its Schwann cells that are kind of making the myelin sheaths here. And we're in the PNS, so that means this is more vulnerable. This could be in your arms or your legs, and it's like, oh my God, I got cut or something or whatever. You've got some damage to your body. Ooh, and the axon got severed. The body has ability to try to repair this and make it functional again. So basically what happens if you sever an axon on one of these PNS, one of these nerves, basically the downstream axon is going to die and the Schwann cells are actually going to kind of reconfigure, kind of clean up the mess and kind of make a tunnel that helps this thing regrow and hopefully find its original target. So the Schwann cells are important for helping repair damage by kind of cleaning up the part that's been hurt and by creating a pathway for the axon to regrow and find its destination. Obviously, this is going to be more likely to succeed if there's a clean cut and you don't have like some real like hamburger kind of injury where it's, they're getting all confused about where's my path back home. But yeah, you can have damage and, these grow pretty slow. So it could be like a month later, all of a sudden, like, oh my God, I'm starting to feel stuff in my hand again. So Schwann cells making myelin sheaths, also insulating or keeping non-myelinated fibers from short circuiting on each other, and also playing a role in repair of damage.
I have another question.
Uh-huh.
Where would multiple sclerosis be involved? Is it more in the CNS?
That's in the central nervous system more.
Oh, okay.
But the oligodendrocytes are also making myelin. This is more of an anatomy thing. They are a little different in that one oligodendrocyte can actually reach out and be involved in many different myelin sheaths on different neurons. So it's not the same where you have one cell per sheath. Here you can have one cell and it's actually playing a role in many myelin sheaths on different neurons. But again, for our class, that's kind of irrelevant. You don't need to really pay much attention to that. You should know, though, I'd feel completely fine asking which are the glial cells that are responsible for making myelin sheaths. You do need to know that. So these are the cells that make myelin sheaths. And then there's some more. So let's continue. All right, so we just took care of these. In the PNS, the only other one that exists, and we don't even need it, you should know they exist, but not much more beyond that, satellite cells. Now I'm wondering, I spelled it right. Satellite cells, you know, they get their name because if you look at kind of neurons, if these are my neurons, the satellite cells are all around. You know, kind of like little moons orbiting around a planet or something. What I should mention, in general, glial cells are smaller and more numerous than neurons.
Are oligodendrocytes, Schwann cells, and satellite cells, those are all glial cells?
Correct. Everything we're talking about that's not a neuron are glial cells. So, you know, what do satellite cells do? People get very vague. Like, they seem to be like kind of cushioning and providing metabolic support. You know, I think because they're not as sexy as the cells that you find in the brain, people don't, yeah, maybe people have found more about them in the recent past, but in general, they don't get a lot of love. So I can't really tell you much more about satellite cells here, other than they seem to be, they're important because they are kind of filling the space around the neurons. But we know a lot more about what the glial cells do in the CNS. So in the CNS, you have a few cells that you really do need to know about. We need to know about astrocytes. Astrocytes, they get their name because they have little processes that come off that make them look like little stars. You know, like an asterisk is like a thing with little stars, or astronomy, like studying the stars. So astrocytes, little star-like cells. These play a number of important roles. One thing they do is they interact with all of the capillaries coming into the brain tissue and help create the blood-brain barrier. So the blood-brain barrier is something you should be familiar with. The basic idea is most capillaries are really permeable, right? I mean, we're going to talk much more about this when we do the cardiovascular system, but again, you already have a basic sense of the role. What is the role of the cardiovascular system?
Transfer nutrients and oxygen and waste.
Yeah, it's where you kind of deliver nutrients and oxygen to the tissues, pick up wastes, pick up hormones, deliver. So in general, capillaries are really kind of leaky, permeable. Even the normal capillaries are pretty, they're kind of designed to make it easy for things to come in and out, because that's the job, is to deliver and pick stuff up. However, in the brain, you got to be way more careful about what exactly gets out into the space between the neurons, because if you have something that acts like a neurotransmitter, for instance, like, we saw adenosine. Adenosine in a normal cell would just be like, I don't know, some building block to make ATP or something. But in the brain, it's a neurotransmitter. A lot of things that you might eat from your diet, if they get just kind of loose up in your brain tissue, might be bonding to receptors as, agonists and stuff like that. So there's this very tightly controlled boundary between the bloodstream and the tissue space around the neurons. That's the blood-brain barrier. It makes it really difficult to design therapeutic drugs that are going to be effective in the brain. You can have a drug that might be the perfect drug if only you could get it in the brain tissue, but if you take it orally or inject it, it never gets across the blood-brain barrier. So like people who design CNS therapeutics have to really not just come up with a drug that's going to be effective, but something that can cross the blood-brain barrier and actually get to where it needs to go. Worst case, if you have some drug you need in the brain, like some antibiotic or something for some infection that is going on, they might actually have to drill a hole and get it in there manually, because it won't go in there just by crossing the bloodstream like most drugs get into your tissue. So blood-brain barrier. We'll see one other barrier the very last week of class, when we get to the reproductive system. There's also a blood-testis barrier. The permeability of the capillaries going into the male gonads, the testes, have also this kind of hyper control over what goes in and out for a number of reasons we'll talk about much further down the road. But the blood-brain barrier. What else? Astrocytes, metabolic support for the neurons. These are the things that are transferring sugars and stuff to the neurons to keep them going. So these are critical in making sure that the neurons are getting all the energy they need. These are also doing, maintain extracellular milieu. Right, as we have seen, the voltages across the membrane are critically dependent on ion concentrations. So these astrocytes are making sure the ion concentrations aren't off. They're also making sure that you don't have too much excess neurotransmitter floating around that has been diffusing out of synapses and might be getting into some other synapse and causing problems or something. And the more people have learned about these things, the more they've realized that they're not just kind of sitting in the wings, just, passively helping the neurons do their jobs. They have slow signaling among themselves. They actually talk to each other, not in the super fast action potentials that we saw in neurons, but in these slower voltage changes. They seem to be important for kind of changing the baseline conditions in different parts of these neural circuits by having more or less neurotransmitter floating around. So they're complicated. The more people have looked at these astrocytes, the more they actually do seem to be playing an active role in kind of how the brain functions beyond just, being the custodians and making sure everything's all clean. But let's leave that there. I'll just say astrocytes, blood-brain barrier, metabolic support, extracellular milieu, and they also seem to be doing something more as well. So you can just leave that for another day. Astrocytes.
So are astrocytes the most abundant glial cell?
I'm not, oligodendrocytes are pretty abundant too, because all the axons in the CNS are myelinated. So they're both pretty abundant. I'm not even sure of how many one versus the other.
Okay, thank
you. There are two more CNS glial cells that you need to know. So we had oligodendrocytes, astrocytes. Now we're going to talk about ependymal cells. These line hollow spaces. Again, if you've been in anatomy, you know that the brain has hollow spaces. So it's a spinal cord. The spinal cord has a big hollow canal going through it. The brain has all the ventricles and aqueducts and stuff in there, and all this cerebrospinal fluid flowing around inside the brain. The ependymal cells are lining those hollow spaces. Ependymal cells have little cilia that sweep back and forth to help keep the cerebrospinal fluid flowing around. We're also going to talk about them, particularly when we get to the choroid plexuses. They work with the capillaries to help actually create the CSF and make sure it has its appropriate composition. So ependymal cells are we'll see them in a little bit as we finally introduce the brain and introduce the ventricles and the flow of cerebrospinal fluid within the brain and outside of the brain as well. And then finally, there is the microglia, which I'm just going to mention briefly here. These are phagocytes. So the brain is one of the places we could say it's immunologically privileged. We're going to talk a bunch about the immune system and all the different things that go on in the immune system. And that's referring to most of your body and how the immune system works in most of your tissues. But the CNS does not have the normal immune responses that the rest of your body has. A big part of normal immune response is inflammation. If you have inflammation inside your skull, you're in trouble because there's no place to swell to. You're gonna shove your brainstem out the bottom of your skull through the foramen magnum and die. So there's a lot of these microglia, which are these kind of little specialized immune cells of the CNS. And their job is to kind of eat things, clean stuff up, be on patrol. But they are the kind of special forces of your immune system that patrol around in your CNS. So, they seem to play roles as well in kind of cleaning stuff up, pruning things. We're going to talk more about kind of all the wiring of the nervous system, right? I mean, you're born with, twice as many neurons as you end up with as an adult. So a lot of that kind of raw material is not used. You wire things up, and as you wire things up, you kind of get the mature circuits going, and then you have to kind of clean up the stuff you're not using. So these are the glial cells. Again, we are going to be spending time looking at all of them little bits, but most of it we're going to talk about are neurons. So glial cells. So finally, we are gonna, I think we're ready to introduce the brain. We're gonna start with the CNS, the brain and the spinal cord. Let me find, I'm gonna, let's see here. So the brain has a lot going on. We're going to talk about lots of different things about kind of more of just the physiology that kind of keeps it alive, you know, the kind of the cerebrospinal fluid and the blood flow and all that. But then we're going to spend a lot of time looking at kind of what it's doing in terms of like the neural circuitry and processing. Oh, here I got it. So I'm gonna share. You know, here is what is going on here. So here we have the skull, and if we get rid of the skull, there's the brain. And if you look at the brain, it is kind of ridiculous in terms of this big folded up mess, right? And it's really kind of hard to understand what's going on if you look at it, because like all this stuff on the outside, that's actually all one part, the cerebral hemisphere that actually has folded around and ended up on top of all this other stuff that is more at the core of it. And it doesn't really give you a sense of the flow of information of like, how are these different, we're going to talk about the thalamus, the hypothalamus, the pons, the medulla, you know, and if you look at the brain just as this thing that looks like kind of a big walnut, you don't really get a sense of that. So I find it is useful to start at the beginning of the development of the brain and kind of look at how it develops. And that way you can see like, oh, this is what it looked like. And then as it grew more and more, those cerebral hemispheres ended up getting really big. And all these folds are because they got all wrinkled up as this surface area, the size of a tablecloth, all got shoved into your skull, but then also kind of obscured a lot of the other stuff that is underneath all of that. So what we're going to do is kind of think about the brain starting from you're just a few weeks post-conception. There was like a sperm, and there was an egg, you know, and then there was this moment of fertilization. There's a zygote, the miracle of new life. And then, and if you remember from biology, there's cell starts dividing, you're a little blastula. Then it infolds and you're a gastrula. And like a few weeks into this all, you start becoming a little embryonic disc. This would be from top. this would be kind of from a side view. You're a frisbee. You're basically a frisbee. And basically around by about a month into development, the top of this thing that you all remember, hopefully you remember from your, what are the 3 germ layers? I know everybody took Bio 110. What are the 3 germ layers?
Endoderm.
The endoderm, the
Mesoderm, ectoderm.
ectoderm.
Mesoderm.
Yeah, so the mesoderm is the middle. The ectoderm, the outer one, folds in. And first there's a groove, and then it actually makes a tube, this neural tube. So again, from the top of our frisbee, it's going to look like a tube, and it's going to have 3 main swellings on the top. So this is this dorsal hollow nerve tube. And the different parts of this thing are going to continue through development to become the different parts of the CNS, right? This is going to become my forebrain and the midbrain and the hindbrain. And all the rest of this is going to become my spinal cord. Things to remember is hollow. So inside of this thing are these hollow spaces. And through development, those hollow spaces are going to stay hollow, although they're going to get very distorted as things get all folded and weird. But, you know, at the end, right now, you sitting in front of your computer has a hollow canal through your spinal cord. It has hollow little caverns, caves within your cerebrum and within between your thalami and, you know, between your cerebellum and your pons that are all filled with this cerebrospinal fluid that we're going to talk about. So this hollow part is important to remember. So what we're going to do right now is look a little more detail about what is the adult structures that end up arising from these original 3 swellings. This, you know, again, in anatomy, it was the prosencephalon, the mesencephalon, the rhombencephalon. For, you know, physio, you could just use the more kind of easy vernacular for mid and hindbrain. So let's look at that. So the forebrain. The forebrain is going to turn into a lot. It's going to turn into 2 separate parts. It's going to turn into what we call the diencephalon. Which are the thalami. So there's 2 thalami. So it's one thalamus and another thalamus on either side. It's going to be the hypothalamus, the epithalamus, people do use the word diencephalon to refer to this level of the brain. So you should know that term. And then above that, we're going to get the cerebral hemispheres. So cerebrum, sometimes they call them cerebral hemispheres. Again, these are the 2 big kind of sides of the walnut that you see. These things overgrow and end up basically overlapping most of the other stuff. So it's harder to see the other parts of the brain because these cerebral hemispheres are so kind of overdeveloped. And they have all these little wrinkles. The wrinkles If you have like a ridge, it's called a gyrus. If you have a valley, it's called a sulcus. You know, those just happen because you've got all of that surface area trying to get all kind of shoved into your skull. So you end up getting all these wrinkles on it. All right, so, and meanwhile, again, the hollow space is maintained. We're gonna have like the lateral ventricles, the foramen of Monroe, the third ventricle. So this is supposed to just be these hollow spaces inside. So as all this is happening, remember that there is a hollow cave that is now getting more kind of convoluted because everything is kind of growing and expanding. The midbrain, let's do the midbrain now. You know, the midbrain usually just ends up being called the midbrain. We're going to talk about different parts of the midbrain later. It's going to include things like the superior and inferior colliculi and the substantia nigra and stuff like that. We will get back to the midbrain. The midbrain also has hollow spaces. The aqueduct of Sylvius is running through the midbrain. And then finally, the hindbrain, or the rhombencephalon here. The hindbrain is going to become, again, if you're in anatomy, it was the metencephalon and the myelencephalon, but we're just going to say that's going to become the pons. And the medulla oblongata. Again, we're going to talk about these in more detail later. Collectively, the midbrain, the pons, and the medulla, we call the brainstem. So the brainstem is basically the midbrain, the pons, and the medulla. You know, and again, it's often hard to really see them much when you have just a full brain you're carrying around in your hand because the cerebral hemispheres are overgrown and fold down over all of this. The brainstem is going to be really important for a lot of like kind of the core functioning, keeping you alive, making sure that you keep breathing and your blood pressure stays stable. And, you know, often people talk about the, you know, the reptilian brain, like this is where you're going to have like coughing and vomiting, right? It doesn't matter if you are a gecko or a human, if something's stuck in your airways, you got to have some reflexes to like get it out so you don't suffocate. So a lot of that kind of core stuff to make sure that you stay alive are down here in the brainstem. Whereas once you get up into the you know, cerebral hemispheres up here, we're going to be talking more about like your personality or trying to figure out interpreting like what does this information mean or something. So whereas again, when you get down here, it's often more like I'm poisoned, I need to vomit. So let's see what, and then one other part of the brain that comes from the hindbrain, which you don't see on this picture, is the cerebellum. which actually kind of hangs off the back of the pons. And again, still we have all of this hollow spaces continuing on. The hollow space continues on, again, continuing as a canal in the spinal cord. The cerebellum, let me go into another page. So I've kind of drawn the cerebral hemispheres, the diencephalon, the midbrain, the pons, and the medulla. The cerebellum kind of hangs off the back of the pons. If I'm looking at the brain from the side, the cerebellum is hanging off here, just underneath the cerebrum. So does that make sense? The cerebellum is hanging off the back of the brainstem behind the pons there. It's going to be important for doing kind of coordinating smooth motor movements and stuff. We'll talk more about the cerebellum later. You know, and it's connected to the brainstem through these axon tracts, right? The cerebral, the cerebellar peduncles and stuff. So it is connected to the all the rest of the circuitry of the brain through a variety of axon tracts. All right, so that is this kind of big picture of how the brain develops. What I'm going to do now is kind of put the brain in the context of the skull, and then add the different membranes that surround it and talk about the cerebrospinal fluid flow. And again, so for that, you have to have a basic sense of just the brain's, what it looks like. And then you have to know about the idea that there are these hollow spaces inside. And then we're going to see there are a variety of membranes that surround the brain as well. We're going to draw this all more neatly, and you'll get to see how this all fits together. And then kind of once we finally kind of get all of this together, we can then finally kind of start introducing like, what does it do? You know, so we'll talk more about kind of the functioning of the brain and processing of information and all that. But let's continue. So I'm going to draw the brain again as if you had a kind of a coronal section, if you cut somebody's face off, basically. So midbrain, pons, medulla. Okay. I'm going to draw the hollow spaces. Again, for this class, you do not need to know the names of all these. If you were in anatomy, you remember I'm drawing the lateral ventricles. These lateral ventricles are the hollow spaces within the cerebral hemispheres. The third ventricle is the hollow space between the thalami. The aqueduct of the midbrain, or aqueduct of Sylvius, is going down through the midbrain. The fourth ventricle is here between the cerebellum and the pons. The central canal of the spinal cord continues down. But for your purposes, you just need to know these are all hollow spaces. hollow fluid filled. The fluid that is filled in those hollow spaces is cerebrospinal fluid. CSF. So we should talk about CSF slightly. CSF is a lot like plasma. It's made by these structures within these ventricles, these hollow spaces called choroid plexuses. So let me talk about what is a choroid plexus briefly. So the basic idea, if you want to make some fluid, one of the most common ways is just to use a leaky capillary. If I have some capillaries here, and we're going to talk more about these later. Special leaky capillaries called fenestrated capillaries. I'm just going to say leaky capillary right now. What kind of cells are covering everything in the hollow places in the brain?
Ependymal cell.
Exactly. So now we have the ependymal cells around all of these. So controlled by both the kind of leaky properties of the capillary but then kind of moderated by these ependymal cells, you basically have fluid leaking out here. Right, I got blood coming here with plasma. Plasma is like 92% water with a bunch, dissolved, salts and stuff. The cells don't leave. Leaky capillaries don't mean the cells leak out, but the water and kind of dissolved solutes leak out. But then the ependymal cells can control the exact composition of it. So the CSF is kind of like modified plasma. Right? It's basically the plasma from the bloodstream is leaving, being modified by the ependymal cells to adjust its composition, and that's CSF. So you basically have these little things that are constantly leaking, constantly making CSF. They are located in a few different areas inside the brain. The choroid plexuses, there's a choroid plexus in both either lateral ventricle. There's a choroid plexus on the roof of the third ventricle. There's a choroid plexus in the fourth ventricle. So those choroid plexuses are sitting there inside the ventricles, constantly making CSF. Again, all of these hollow spaces are lined with ependymal cells. So those ependymal cells have little cilia that are kind of helping circulate it around, keep it flowing. But it's constantly being produced. You produce something like about a half a cup every, like, I don't know, 5 to 8 hours. So you need to we need to drain it as well, or else our brain is going to inflate and explode, which would not be ugly. So we have to have a more complicated picture now. So this is the brain and the CSF that's forming inside. Now I'm going to start adding on some of the other parts that enclose the brain that are also going to play a role in the flow of CSF, because the CSF is constantly produced. It's going to have to go someplace. So we need more of the overall anatomy to see where it goes. All right, so the meninges, we're going to talk about the meninges now. So meninges are the layers of different membranes that cover the brain, and there's 3 of them. From the inside to the outside, there's the Pia Mater, which is we'll talk again, I'll talk about it in more detail in just a few moments. Then the one outside of that is the arachnoid layer. And then the outermost layer is the Dura Mater. So we are going to look at each of these now in turn and then see how they ultimately fit into our story with the CSF as well. So Pia Mater. Pia Mater is a very fine connective tissue layer that is on the actual brain tissue. If you just look at a brain that's been pulled out, you don't even see it because it's really fine and it follows every little fold and nook and cranny of the brain tissue. Pia means faithful. So it goes up and down on every little thing. So if I can make it green. So I'm going to say Pia Mater is green. Then it's going to be on top of the brain tissue and follow every little, sulcus and gyrus up and down. So this is all my Pia Mater. It plays a little more of a, obvious role when you get down into anchoring the spinal cord within the spinal column. You have the little denticulate ligaments, you have the filum terminale. Those are actually bits of Pia Mater that extend out where the fibers help anchor your spinal cord and keep the spinal cord from riding up your spinal column. But in general, if you just look at a brain, it's not an obvious thing to see. The arachnoid layer comes next. Arachnoid layer gets its name because it's got little webby struts, kind of like a spider web. So the arachnoid layer has these little spider webby struts. That anchor on the pia. And that leaves a space underneath where CSF can flow. So underneath, there's going to be what we call the subarachnoid space. We will get back to the subarachnoid space in a few moments, but that is basically, in fact, let's just talk about it now. The CSF actually drains into the subarachnoid space through these little these little holes in the fourth ventricle. So there's the median and lateral apertures here. Maybe I'll type because it'll be so basically you have these little connecting, they're also the foramina of Magendie and Luschka is the old name for them, or you can call them the median and lateral apertures. They basically allow the CSF, which is being constantly produced and constantly produced inside these choroid plexuses within the ventricles, but then the CSF gets to flow outside, and now it's also in the space, in the subarachnoid space underneath the arachnoid, but outside of the brain. So the basic idea here is that the brain is floating in salt water, right? The brain has, CSF is basically salt water. It's like plasma. So the brain has salt water inside and outside. It's like, imagine you've like filled your bath and loaded up a bunch of Epsom salts and you're just kind of sitting in there and you're pretty weightless. Like the brain weighs a few pounds and it's really soft. Like the brain that you hold, if you're in anatomy, that's been fixed with formaldehyde and cross-linked all the proteins, makes it feel pretty tough. But I've never actually seen a total fresh brain that hasn't been fixed at all, but supposedly it's the consistency of kind of like cold oatmeal or something. It's really not, or soft tofu, something like that. So it's kind of delicate. So you have it floating in salt water, so it's pretty much weightless inside your skull. Again, imagine you're in the bath and you even kind of forget you've got a body. You're just like, this is so nice. All the worries of the world are fading away. And then, I'm sure most of you, I know it's kind of fun. Sometimes you kick out the little, what do you call it? The little stopper and let the water go, but just stay in as the water goes lower and lower and lower and lower and lower. And then you just start feeling your weight. And all of a sudden you're like, holy crap, I weigh 150 pounds plastered here on the bottom of the tub. I thought I was like floating. It's kind of the same thing with your brain and your skull. That we're going to talk about like spinal taps. One of the things, after you remove some of the CSF for a diagnostic procedure, people have to lie down flat for hours and let everything recover. If they try to sit up too soon, they get like just horrible, excruciating headaches and things like that. So the brain is kind of floating there. So CSF is being produced in the choroid plexuses, leaving via these lateral and median apertures into the subarachnoid space. Obviously, we still got to go someplace from there. We still have to talk about the Dura Mater. Let's do the Dura Mater now. So the Dura Mater, Dura means like the hard mother. So the Dura Mater is going to be, it's actually dense irregular connective tissue. It's basically like leather. It's really tough. again, if you've been in anatomy, you've seen it. It looks like a bathing cap, like a swim cap on top of the brain. It's really, it's hard. It's tough. So I'm going to draw it. And particularly in the skull, it actually has two layers that ends up providing the possibility for these little passageways. They're called dural sinuses, which you do need to know about. So the Dura Mater has little canals inside. The Dural Sinuses. So like this one here that I've just drawn, it's like the superior sagittal sinus, if you remember from your anatomy. And there are little valves that drain CSF from the subarachnoid space into those dural sinuses. Now this is getting messy here. These are my, oh, let me get a, so the arachnoid villi drain CSF from subarachnoid space into the dural sinuses. This is, it's really not, I'm realizing I am being not very legible. So I'm going to just type that out using the type tool. Again, the dural sinuses are just these little canals inside the Dura Mater, and these then drain into venous circulation via the jugular vein. So all of those dural sinuses, all of these canals that I was talking about, these all ultimately drain into the jugular vein. So if you think about it, CSF started out as basically plasma kind of leaking out of the bloodstream, you know, slightly modified within the ventricles. That CSF is then flowing around inside the ventricles of the brain, then goes out through the apertures into the subarachnoid space. And then from the subarachnoid space goes into the dural sinuses. From the dural sinuses, it goes back into the jugular vein, into the venous circulation. So it's made a full loop. That fluid is now part of your bloodstream again. So it's constantly being produced and constantly being drained. Does that make sense? Let me go into images. Like if I go, I'm going to share screen. Like hydrocephaly, like if that drainage is not working properly, especially if you are like, in development, if you remember your brain is inside your skull, but your skull bones are not actually ossified yet when you're still a fetus, right? A baby is born, their skull's all squishy. So if you have that drainage blocked, the brain is going to start inflating like a balloon. And because these skull bones are not actually bone yet, they're actually just this kind of membranes that are undergoing membranous ossification or intramembranous. So let's take a look here. And you can see these like kind of horrible pictures, right? Where the brain is inflating. Those ventricles are not draining properly, and instead the brain is inflating. And, this basic idea of let me see, I want this bigger. Oh, I can't do it. But anyway, the basic idea is you can put a shunt in there, and then you can get the fluid to drain. So even though this looks horrible, it's not necessarily like, doesn't mean the kid's not going to be okay ultimately. They can put a little shunt, a drain in there to restore proper flow so things can develop properly. So again, normal brain, those ventricles exist, but in hydrocephalus, those hollow spaces start inflating and becoming kind of ballooning out and inflating the whole brain. So just kind of putting that out there. What else? Last thing I should mention before, or actually a couple of things I'll mention before we take our break. This thing here is illustrating that the Dural Sinus. So in this picture here, I'm going to go to annotate. This is the Dural Sinus. This is that tunnel within the Dura Mater. Again, the Dura Mater actually has kind of these two layers. This space under here is the subarachnoid space. And then this thing here, these are the arachnoid villi. And you can see they kind of make these little like buttons where they kind of push into the dural sinus. So sometimes they're also called arachnoid granulations, because if you just kind of look, it looks like kind of little bumps, like little granules where the arachnoid villi are. So they can be called arachnoid villi. They can also be called arachnoid granulations. But basically the idea is cerebrospinal fluid is in here, goes through these, and now is in here. And once it's in here, it goes off to the jugular vein and joins up into venous circulation. So again, you should understand the basics of cerebrospinal flow. Again, being produced constantly, being drained constantly. The last thing I will do is talk about spinal taps. So let's put the brain and the spinal cord. Here's the brain, here's the spinal cord. And then the spinal cord has all these nerves that come off to go to the parts of the body. The spinal cord ends around lumbar 1 or lumbar 2, like the level of lumbar vertebrae 1 or 2. But if you look at the meninges that cover this, the meninges these are, so this is like the Dura Mater. The Dura Mater actually goes all the way down to the bottom of your spinal cord, down to the coccyx there. So what that means is you've got this space here that is below the spinal cord, but is still inside the subarachnoid space here. So it's got CSF. There's CSF underneath all of this, obviously. And what you can do, if you need to get a sample of the CSF, let's say you want to make sure that there's no blood in there or no white blood cells or no bacteria or something. There's a lot of reasons why you're going to want to check the composition of the CSF. And you don't want to drill a hole into their skull and stick a needle down through their brain tissue into the ventricles. You can instead, again, we have the, the vertebrae that are around that make the spinal canal. You can take a big needle and you can jam it through the intervertebral disc, poke through the Dura Mater, get into there, because as long as you're below lumbar 2, you know that you're safe, that you're not going to actually nick the spinal cord and damage the person's CNS. And you can suck up a little bit of that fluid and analyze it. So this is called a lumbar puncture. Also called a spinal tap. So it's a way to get a sample of the CSF to analyze, minimally invasive, especially if you consider the idea of sticking a needle into the middle of your brain. Like I mentioned previously, you need to kind of lie on your back and let things kind of heal and re-equilibrate before you sit up. Supposedly, if you sit up too soon, like before a few hours, you can get excruciating headaches. I have heard of, actually, I knew somebody where there was kind of continuous leak. He had was having trouble actually getting things to seal permanently. But in general, after a little while, you're kind of good to go again. So that is a spinal tap. It's not just a band or fake. It never was a band, but it's not just the idea of a virtual band.